Hello everyone, welcome to the Drug Channel. In vitro in vivo correlation, commonly referred as IVIVC, is a scientific relationship that links what we measure in the laboratory with what actually happens inside the human body. You must remember that before a drug can be absorbed, it must first release from the dosage form and dissolve, which we study using in vitro dissolution tests. IVIVC attempts to answer a very practical question: can we predict a drug's in vivo performance simply by studying its in vitro dissolution behavior? If such a relationship exists, it becomes a powerful tool in pharmaceutical development. From a conceptual standpoint, IVIVC established a mathematical or point to point relationship between fraction of drug dissolved in vitro and fraction of drug absorbed in vivo. This is especially relevant for modified release formulations where the drug is released slowly over time and the dissolution profile directly governs the absorption pattern. When a strong IV-IVC is achieved, changes in formulation, manufacturing process, or scale up can be evaluated using dissolution studies alone without repeatedly consuming costly and time consuming human bioavailability studies. IV-IVC is commonly classified into different levels, with level A being the most informative and preferred by regulatory authorities. Level A IV-IVC represents a direct point-to-point correlation between in vitro dissolution and in vivo absorption profiles across time. Lower levels such as level B and C provides less detailed relationship and are generally considered supportive rather than definitive. In regulatory practice, level A IV-IVC is the gold standard because it allows reliable prediction of plasma concentration time profiles from dissolution data. IV-IVC play a critical role in formulation optimization, post-approval changes, and biowaivers. Regulatory agencies adopt well-established IV-IVC models to justify changes in formulation composition, manufacturing site, or process without repeating in vivo bioequivalent studies. In summary, IV-IVC acts as a bridge between the laboratory and the clinic, establishing scientists to translate simple dissolution experiments into a meaningful predictions of drug behavior in humans. As pharmaceutical scientists, understanding IV-IVC empowers you to design a better dosage forms, reduce development cost, and ensure consistent therapeutic performance.